So, dear students, we shall continue the chapter Biotechnology, the part first. Question and answers, the discussion. In this video, the video number 3. Okay. The first question is, name a pathogen virus which can be disarmed and used to deliver genes into animal cells. The virus retrovirus is used as a pathogen virus which can disarm and used to deliver the genes into animal cells. The answer is retrovirus. Retrovirus are disarmed before using to deliver desired genes into animal cells. Why? What is the reason why we should disarm? Means because the virus, the retrovirus, have the ability to transform the normal cells into cancerous cells. Means the retrovirus can cause cancer. That is why the virus must be disarmed before using to deliver the desired genes. Next question. What is competent host? Spelling tappagide. Competent host. The host, in which the desired gene is introduced. The organism in which desired gene is introduced is called the host. What is competent host? The host is made competent to receive the rDNA. In rDNA technology is called competent host. It means the host is made competent to receive the rDNA. So there are different methods are there to make the host competent, that you know. Next, very important for the examination, what is microinjection? We may expect in the examination. The direct injection of desired gene into the nucleus of an animal cell using microsyringe or needle, fine needle, is called microinjection. So this is one of the methods by which we can introduce a desired gene. Okay. Next question. Mention the enzyme used to isolate DNA from bacterium or bacterial cell. This is one question ಆಗುತ್ತೆ. ಅದೇ ರೀತಿ ಇನ್ನೊಂದು ಕ್ವಶನ್ನು ಪ್ಲಾಂಟ್ ಸೆಲ್. Similarly, one more question for 1 mark. Fungal cell. First bacteriumಗೆ ಆನ್ಸರು, the enzyme used to isolate DNA from bacterial cell is Lysozyme Enzyme. Very important, Lysozyme Enzyme. It dissolves the bacterial cell wall or cell membrane. Then in the plants, the enzyme cellulase is used to isolate the DNA. Cellulase digests the cell wall of plant cell. Suppose in case of fungi, that is fungal cells, the chitinase enzyme is used. So lysozyme for bacterial cell, cellulase for plant cell, chitinase for fungal cells. Okay. Next. Why chilled ethanol is used in DNA isolation? See, while isolating the DNA from a cell, at the end, chilled ethanol is added. When we add chilled ethanol, the DNA gets precipitated and it occurs in the form of a thread. We call spooling anta ಕರಿತೀವಿ ಇದನ್ನು. So that's all. Well, now expand PCR. P means polymerase. C chain or reaction. Polymerase chain reaction. What is PCR? The process or the method of making many copies of DNA or the gene of interest is called polymerase chain reaction. By polymerase chain reaction, we can produce billions of copies of DNA or the gene. Next question. Name thermostable enzyme used in PCR technique. The enzyme is Tag Polymerase. The name of the enzyme is Tag Polymerase. Name the bacterium from which the thermostable Tag Polymerase is obtained. The name of the bacterium or bacterial name is Thermus aquaticus. These 9 and 10 questions are very important questions for the examination. Next, we shall pass on to the next slide. What is the use of Tag Polymerase Enzyme? Just now you have seen the Tag Polymerase Enzyme is a temperature resistant enzyme. It will not denature in high temperature. Okay, it is used in PCR. Why tag polymerase is preferred over the normal DNA polymerase in PCR? Because tag polymerase enzyme is high temperature resistant enzyme. It will not denature in high temperature. But normal DNA polymerase, it will denature in high temperature. That is why it is preferred. Next question. What is recombinant protein? The protein coded by recombinant DNA is called recombinant protein. Okay. Next, very important question. What is bioreactor? Bioreactor is an apparatus for growing organisms like yeast, bacterial, animal cells, in which the various biochemical reaction or biological reaction takes place, or the process is carried out under controlled conditions on a large or industrial scale. Okay, very important question. Next, one more important question is there. What is downstream processing? DSP. The process of purification and separation of products produced from rDNA in rDNA technology is called downstream processing. Okay, so that's all about one mark question. Now I am going to discuss one 3-mark question. 3-mark question. Write the function of restriction enzymes. How the enzymes function. Okay, now we shall see. Restriction endonucleases are the enzymes functions by inspecting the length of the DNA molecule or the sequence. It means first it inspects the DNA molecule. Secondly, the restriction enzymes finds its specific recognition sequence. I already told you about recognition sequence. 6 bases on DNA molecule forms recognition sequence. So now the enzyme, before functioning, it identifies the specific recognition sequence. Then the enzyme binds to the recognition sequence of DNA and cut 2 strands of DNA at specific points. That is why called molecular scissors, where it cuts in the sugar phosphate backbone. Each restriction enzyme recognizes specific palindromic DNA sequences. I will give you an example. ಒಂದೊಂದು ಎಂಜೈಮು ಒಂದೊಂದು ಪ್ಯಾಲೆಂಡ್ರೋಮಿಕ್ ಡಿ ಎನ್ ಎನ ಐಡೆಂಟಿಫೈ ಮಾಡಿಕೊಳ್ಳುತ್ತೆ ಕಟ್ ಮಾಡೋದಕ್ಕಿಂತ ಮುಂಚೆ. Look here now. What is this palindromic DNA? Palindromic sequence on 2 strands of DNA in both the direction. ಇದು ಸ್ವಲ್ಪ ಮಿಸ್ಟೇಕ್ ಆಗಿದೆ ಇಲ್ಲಿ. Palindromic DNA reads same sequence of nucleotides on 2 strands of DNA in both the direction, that is in forward and backward direction. ಉದಾಹರಣೆಗೆ ಯಾವ ಥರ ಅಂದರೆ, it is just like reading a word madam from left to right. Madam. The word, if you read from right to left, madam. Similarly, palindromic DNA is GAATTC, one of the strand in 5' 3' direction. In the another direction, in another strand, 3' and 5'. You can see now GAATTC, backward direction. How the restriction enzyme function means, the enzyme cut the DNA at a particular point. For example, ECOR first, see the diagram. It cut the palindromic DNA between G and A. Observe carefully. The enzyme cut the palindromic DNA sequence between G and A in both the strands. So after cutting, sticky ends are formed in both the strands. Sticky ends. This is how the enzyme function. See the diagram carefully. Okay. Cut the DNA in the middle and produce single strand at the end, and these ends are called sticky ends. I told you. What is the use of sticky ends? These ends helps in binding other DNA strand by the enzyme DNA ligase. Okay, so that's all. One 3-mark question. Next, one more 3-mark question is also there. Write diagram of recombinant DNA. Now you can see the diagram. Practice this diagram, it's very important. See, foreign DNA, left side. See, vector DNA on the right side. Now both the DNAs are treated with the restriction enzyme, that is arrow mark. It will cut DNA and thus desired gene is obtained from the foreign DNA. Here the same enzyme cut the plasmid. In both the cases stick-ends are forming. Then both are linked by adding the enzyme DNA ligase. Now RDNA is formed. Circle iddangirodu RDNA. So this is called recombinant DNA because it contains 2 DNAs. Then the recombinant DNA na host cell ಒಳಗಡೆ ಹಾಕ್ತಾ ಇದೀವಿ. Host is equalized. Now the host cells are multiplied ಮಾಡ್ತಾ ಇದ್ವಿ. That is cloning ಮಾಡ್ತಾ ಇದ್ವಿ ಅಂತ ಹೇಳ್ಬೋದು ಬೇಕಾದ್ರೆ. So the diagram, you practice diagram, very important diagram for the examination. Next, ಇದನ್ನೇ 5-mark questions ಇರುತ್ತೆ. Explain RDNA technology or recombinant DNA technology. ಇದೇ ಡಯಾಗ್ರಾಮನ್ನು ಬರೀಬೇಕು. Explain the process of recombinant DNA technology. Process of 5-mark question. Process of recombinant DNA technology or RDNA technology involves following steps. First step is isolation of DNA. Second, cutting of DNA by restriction endonuclease and isolation of, that is, separation of desired or alien gene. Second, suppose the desired gene is very little, ಸ್ವಲ್ಪ ಇತ್ತು ಅಂದರೆ amplification of the gene may be done by using PCR. Next, insertion of desired gene into vector or plasmid to get RDNA. The next step is insertion of RDNA into the host cell. Last one, culturing the host cells and obtaining the foreign gene product. So these are the 6 steps involved in RDNA technology. Okay? Now you see the diagram. The same diagram we repeated. Practice it. Very important. Practice is very important. Now we shall explain. Isolation of DNA. You know the DNA is present in the cells, that is within the nucleus. In the nucleus, DNA is present in the chromosomes. How to isolate this DNA from the chromosomes? Okay? This DNA is present in the chromosomes, and chromosomes are enclosed by nuclear membrane. Say, you imagine you are isolating the DNA from bacterial cell. How to isolate the DNA? The bacterial cells are treated with the enzyme lysozyme. It dissolves the cell wall. Similarly, if you are isolating the DNA from plant cells, plant cells are protected by cell wall. Cell wall is made up of cellulose. So this cell wall is treated with, or the cell is treated with cellulase enzyme. It will digest the cell wall. You imagine you are isolating the DNA from fungal cells. The fungal cells are treated with chitinase enzyme to digest cell wall. Thus, appropriate enzymes are used to isolate the DNA. The cell not only contains the DNA, also contains RNA, fats, proteins, and carbohydrates, the other chemicals. The fats are dissolved by the enzyme lipase. The carbohydrates present in the cell are disintegrated or dissolved by the enzyme amylase. The RNA present in the cell is denatured by RNA polymerase enzyme. Now, the cell or the DNA is treated with chilled ethanol, or chilled ethanol is added. What happens? Now the DNA precipitates. It occurs in the form of thread-like structure. This process is called spooling. So in this way, the DNA present in bacterial or plant or fungal cells isolated. So this is also for 3-mark question. Explain isolation of DNA. So this is the first step. Now we obtain the DNA molecule. Second step in our DNA technology is, now you can see the diagram of spooling also. Okay, DNA that separates out can be removed by spooling. Now the second step is cutting DNA by restriction endonucleases to get desired alien gene. Purified DNA is treated with restriction endonuclease. For example, ECO or FAST. It cuts DNA into pieces. The DNAs are separated, purified and separated by using the technique called gel electrophoresis. In the previous class, you have studied the gel electrophoresis, and how the DNA is obtained in this process. Amplification of gene by PCR. If the available gene is very little or DNA is little, number of copies made by PCR. That's all. Later you are going to study PCR. That's all the second step. Third step, insertion of desired gene into plasmid to get rDNA. Now the desired gene with sticky ends, plasmid with sticky ends mixed together with enzyme DNA ligase. As you know, the ligase join the desired gene and the plasmid DNA. Then recombinant DNA is formed or rDNA is produced. This is called rDNA because it is having two DNAs. Okay. This technology is called rDNA technology. Next. Now the rDNA is inserted into the host cell. Insertion of rDNA into the host cell. DNA is mixed with E. coli. That is rDNA mixed with E. coli host and treated with the divalent cation of calcium, followed by heat up to 42 degrees Celsius, which make the pore on the cell wall, then placed on ice. So now the rDNA enter into the bacteria, E. coli cell or the host. ಇಲ್ಲೇನು ಮಾಡ್ತೀವಿ, rDNA ಮತ್ತು ಹೋಸ್ಟ್ ಸೆಲ್ ಎರಡು ಮಿಕ್ಸ್ ಮಾಡಿ, ಸೊ ಕ್ಯಾಲ್ಸಿಯಂ ಕ್ಲೋರೈಡ್ನ ನಾವು ಹಾಕ್ತೀವಿ. ಬ್ರೀಫಾಗಿ ಹೀಟ್ ಮಾಡಿದಾಗ 42 ಡಿಗ್ರಿಗೆ ಮತ್ತು ಐಸ್ ಮೇಲೆ ಹಾಕಿದಾಗ ಸೆಲ್ ವಾಲ್ ಮೇಲೆ ಪೋರ್ಸ್ ಫಾರ್ಮೇಷನ್ ಆಗಿ rDNA enters into the cell. Now the host cell contain rDNA. So this is one method of introducing rDNA into the host cell. Other methods are also there. Say for example, in case of animal cell, microinjection is done. In the plant cell, other method, gene gun method is followed. Or it's also called biolistics. And another method is also there, using disarmed retrovirus. The three methods ಈ ಥರ ಇದೆ. Now, host cells. Culturing the host cell and obtaining the desired product. The host cell is multiplied. It means cloned. If the gene coding for protein is incorporated in rDNA, the protein synthesized is called recombinant protein. Okay. For example, if the insulin gene is introduced, insulin is produced. If recombinant protein required in small scale, bacteria are cultured in the lab. You imagine the recombinant protein is required on a large scale, that is industrial scale. Then the bacteria are cultured in bioreactor. Then lastly, by downstream processing, the product is purified. Now the DNA is used, rDNA, this is, okay. So that's all in this video. ಥ್ಯಾಂಕ್ ಯು.